invite Alok to come up and speak to us about gene therapy progress for hemophilia and thalassemia in India, and anything else you'd like to share as well. Do you have, yeah, can you just load that up?
Okay, so thank you, Chandu, thank you, Namita. I'm happy to be here for many reasons, mostly because I was told that there are patients who want to know more about this, and that's something. That's something that. Okay, so keep the lights on. Please keep the lights on. Yeah, thank you. You can keep the lights on. And this light is the one you should cut down. The one on the speaker, please cut down. Just put on the right. Yeah. Hall ka light बढ़ा दीजिए इसको. Yeah, that's fine. I think you can see. There's nothing. So as I see, they're mostly patients probably in the audience. So I will keep the technicality low and try to just go through the broad message that we want to discuss about gene therapy. So gene therapy means replacing or repairing a gene that is not functioning. Okay. Currently, there are two major approaches that are being taken. What is shown on your left is where you have the gene packaged in a vector, a transport vehicle, usually a non-harmful virus, which you can inject directly into the patient's vein, and it will find its way to the right tissue. And this is the approach that is being taken right now for hemophilia mostly. The second approach is where you take out certain cells from the patient. And by the way, I should mention that this approach is also being considered for Duchenne muscular dystrophy. The other approach is where you take out certain cells from the patient's body. You then treat those cells with a vector, again a virus carrying the good gene, which will then infect those cells, and then you may manipulate these cells a little further, expand it if needed, and put it back into the patient's body. And this is the approach that is currently being taken for thalassemia. So this is the approach for hemophilia right now, this is the approach for thalassemia. Now, what the gene does, it gets into the patient's body, it gets into the right kind of cell that will make that protein. We have all the genes in all the cells of the body, but all the cells don't make all the proteins. Specific cells are designed to express specific proteins. So here we have to find a cell where you send the normal gene into the cell that will actually make the protein. So here it has to go to the right cell, and then it makes protein. The protein is then secreted. It may be factor 8 or factor 9, it may be hemoglobin, it may be some other enzyme protein. Okay, and then if you are, your disease is ameliorated, you may be cured depending on how early the treatment is done and how well the protein is expressed. So today there are 4 approved gene therapy products in the world. Starting about 3 or 4 years ago, the first one that was approved was an AAV. So one of the vectors is called adeno-associated virus. It is a harmless virus, and that was approved about 4 years ago for a rare disease called lipoprotein lipase deficiency, where people get disease in the pancreas. And this was in the market for, it still is it was in the market for till about last year. The company had priced it at €1 million, almost nobody bought it. They withdrew it from the market. What is in the market right now in Europe is a lentiviral vector. So this is a lentivirus with a gene for immune deficiency that is faulty in people with severe combined immune deficiency. And this is in the market. It's called Strimbalis. It costs 1 million euro. This is being used. And then last 3 or 4 months in the US, these are 2 gene modified cells. So these are the patient's own immune cells, T cells, that have been gene modified to treat certain cancers. This is for acute leukemia, this is for lymphoma, priced at about 400,000 to 500,000 US dollars. Okay. So with these kind of prices, somebody wrote a comment on the internet and they said these prices are not costs of drugs. These prices are like ransom. It's your money or your life. If you want to, okay? So, we were talking about insurance cover, and in countries where the government or the insurance takes responsibility for citizens' health in a significant way. We also take responsibility on paper, but in practice, it's really not there. It is improving rapidly, but not as much as we would like it to be. So these kind of costs are going to be a challenge in this country, and that is one of the major reasons why we have a focus on developing gene therapy indigenously, because without that, these costs will be impossible for us. So very quickly, everybody, so how many people with hemophilia in this room? I can see just 2 or 3, right? So and they have had opportunity to hear this elsewhere, so I will finish hemophilia very, very rapidly, and we will not worry too much about the details here. So the treatment hemophilia is, for deficiency of 8 or 9, and the treatment is to replace the factor which is missing. And because it is expensive, because it is cumbersome, it is not done properly, and the result is that people land up with frequent bleeding into the joints and muscles, because of which they have a lot of musculoskeletal challenges, which affects their mobility mainly. Now, in 2011, we had the first successful gene therapy reported. Again, what did people do? They took an AAV vector. The adeno-associated virus exists in nature and causes no disease. Why nature made AAV? Maybe they wanted to give man an opportunity to do gene therapy. There is no other function for AAV that anybody has found so far. And it's very, very useful in gene therapy. So this was, 4 or 5 years ago now. In 2014, they reported the updated study. And at that time, 5% expression of factor 9. That was magic. Okay? And people were celebrating, and this was published in December, and so said, Merry Christmas for people with hemophilia B. Now, since then, the world has moved on. And we had so many trials. I showed you this slide last year. I also showed you that there were some problems with some of them. The trial stopped. But the important one was the trial by Spark Therapeutics, where factor 9 now was in the 29-30%, 25 to 30% range. So 5% was not good enough. It's not 25-30%, getting closer to normal. And factor 8 also, there are a huge number of trials, one of which was giving even 100% expression, all AAV based. So this is what's been reported from those 2 trials in the last 2 months, 3 months. Early December, late December 2017. So this is the Spark trial, which shows that about 10 patients with hemophilia B treated, reported in December 2017, are getting factor levels from around 10% to 50-60%. Okay, AAV based. So much research, so much work. Finally, the treatment is an outpatient injection. Okay, you take an injection in your vein, you go home. 4 weeks later, you express protein, and you can be cured, or depending on how much you express. So, if and when it finally becomes approved treatment, the treatment will be as simple as a peripheral vein injection. The same for hemophilia A. So now here in one they were using AAV 8 In this it's AAV5. Okay. Now here what you're seeing is Hemophilia A. Look at the patients who have been treated and their factor levels. Almost in the normal range, many of them. Okay, 50 to 150 is the normal range. So both Hemophilia B, Hemophilia A, this is happening, and these are now, so these were the phase 1-2 trials. You heard today about phase 1-2 trials. Phase 1-2 trial shows it is feasible, it is safe, and it is reasonably effective. Then you do a phase 3, which is, in these diseases we don't really do a comparison because there is really nothing to compare with. But phase 3 just means a larger number so that you have a higher chance of picking up any adverse effects that may come. So that's where it's moving now. Okay. So with that background, gene therapy for hemophilia is there. Is there today. The question is, phase 3 trial starting in about 1 to 2 years. That should finish because recruitment is very fast. The product, you know what the biggest challenge has become in the world? In the US, there are several companies trying to do gene therapy. The biggest challenge is to make the vector. There are no industrial facilities to make enough vector. So in fact, New York Times had an article a few weeks ago saying not enough units to make the vector for gene therapy trials. So nobody had expected, industry would have, if they knew this is going to boom like this, people would have invested in actually making GMP facilities to make enough vector. Nobody expected this success so rapidly. But it's a good thing, it's a good problem to have because, surely they can build facilities to make vector. So keeping all this in mind, we have been, thinking about gene therapy for more than 10 years. But in the last 2, 3, 4 years, it has really gelled together, particularly because there is very strong funding from the Department of Biotechnology of Government of India. So we have a collaboration, as I told you last year, with Emory, with Florida, with us. Emory is making the transgene, Florida is helping us with the vector, and we are doing all the clinical assessment of selecting the right patients and doing the clinical trials. So we actually had a meeting with the hemophilia community here last year to explain what we are doing and the importance of gene therapy. So here to again, not to repeat much of what we said last year, we have selected a particular serotype which we think is better suited. We have made a transgene in collaboration with Emory, which is different from the one that has been reported so far because there will be issues of intellectual property that will come up. And the important thing was to show that this transgene actually functions, that it actually makes factor IX. So what we did was we put this transgene into, so here's the challenge. AAV3, so we have chosen AAV3. I didn't want to get into this detail, but I can't explain this to you without the subsequent data without showing this. So people are using AAV8 and AAV5. I showed you the 2 successful trials. AAV8 and 5 infect mouse tissue also. So in the preclinical study, preparing for the clinical trial, you can put your transgene into AAV5 or 8 and inject a hemophilic mouse, and you will show production of factor 8 or 9, whichever you put in. In the case of AAV3, it doesn't infect mouse, and that is why people had missed it. People had not thought about using it. And but AAV3 has the highest affinity for human liver, much more than the AAV8 or 5. And that is why we are choosing AAV3. But the problem is, if you put the gene into AAV3 and you inject it into the mouse, what will happen? It will not infect anything. So we won't know whether our transgene is working or not working. Can we inject it straight into a human, saying, okay, mouse is not getting infected, what can we do now? Put it in the human. We can't do that, right? So now you have to find a way to, so we did 2 things. We first put the transgene into AAV8 and put it into the mouse, comparing it with the one that was already in clinical trial. And see, this was the range of factor IX expression with that. This is the range of expression with our transgene. So it's making factor IX in the mouse. But that's not good enough. We didn't put it into AAV3. So if you have to put it into AAV3, as I told you last year, what we have to do is to make a special mouse, which is called a humanized mouse with human liver cells. And that's not easy to do. It is done, but it is done at a cost of about $3,000 a mouse. So that's the data we just got about 2 weeks ago, that this transgene in AAV3 in the humanized mouse is giving you factor IX expression in a clinically significant way. So what is remaining to be done now? We're supposed to do the non-human primate studies. This is the first time AAV3 will go into humans, and therefore we would like to make sure that there is no toxicity that is not expected. And so we'll do this, and in parallel we are trying to develop the vector in the GMP. And that's not a simple challenge, because the vector cores are all blocked by trials going on in the US. So that's where we are, but when the vector comes, it should be not too difficult to do the trial at all. And preparing for that trial, we have got approval from our institution, from the DCGI committee in principle of what we are trying to do till we go back with the specifics. One challenge remains. In this room, there are probably 50 people now. Anywhere from 30 to 70% of us are infected with AAV. If we have AAV antibody, we will not be eligible for this trial. Okay? So then, we don't know in our community what is the AAV antibody incidence. So we are doing a large study to evaluate this. We have collected about 500 samples from patients in the south mostly with hemophilia, and we're trying to assess, and the initial data, one slide is missing here. The initial data shows that we have about 50-60% incidence of AAV antibodies. So that's a damper, because many patients may not be eligible for this trial. Now what can we do for them? So our collaborators in Emory, for hemophilia also, they have shown that you can actually use the hematopoietic stem cell, the blood stem cell, to express factor VIII. So they have made a lentiviral vector with factor VIII, put it into a hematopoietic stem cell, and showed that it can make factor VIII. Okay? So this is what is actually ready to go to the clinic. What they did is, they took a mouse, they took the mouse blood stem cell, they put the transgene into the blood stem cell with a lentiviral vector, and then they transplanted this into another mouse that had been irradiated. So now this mouse has got a lentiviral vector stem cell, it is making factor VIII. And what they showed is that they can make a large amount of factor VIII. So this is very different from AAV. You know, the other one I told you, you come to the clinic, you take an injection in your vein, you go home, your gene therapy is over. This is very similar to thalassemia. And the reason we are interested in this for hemophilia is that in a way it paves the ground for thalassemia also. So here, what is done, and I'll skip this, what is done is that you have the patient, you give them a growth factor so that the stem cells come out of the bone marrow, and you collect the stem cells, then you infect the stem cells with the lentivirus vector, which is carrying the factor VIII gene, and then you do an autologous transplant for the hemophilia patient. So that is much more involved. So we have to think very carefully about this. You know, the patients with thalassemia in this room, they are mentally prepared. One day we have to have a transplant, and even our gene therapy will be autologous transplant, otherwise it is an allogeneic transplant. And we'll talk about that in 2 minutes. But hemophilia patients are not thinking about transplant. So it's a new paradigm for them. But we are looking at this, we are actually proposing that we try this, because regardless of the success of AAV gene therapy, those patients who have significant antibodies to AAV will never be eligible for AAV based gene therapy. So they need an option, and this gives an option to the patients with hemophilia who have AAV antibodies. So we'll move now from this to thalassemia, and I don't need to tell you numbers, you're familiar. I only will show the same data that I showed about transplant. Transplant is hugely successful depending on when it is done, success ranging from 70 to 95%. The good thing here is that in the last 1 year, the Coal India Corporate Social Responsibility Project has given more option. It is still not running full steam because they have restricted this project to only those centers which are non-profit, and so only 4 centers in the country are eligible at the moment for doing these transplants, and a total of about, I think, 60-70 patients have benefited so far. The hope was that it will benefit at least 250 to 300, going up to 500 patients in a year. So it needs to be expanded, but and all the issues related to transfusion chelation and transplant we can discuss separately. As I was mentioning during tea, please have a separate session where it is for patients only on thalassemia, where the kind of questions I get asked each time I come here, we can discuss in much more detail, and then you can put up some of that information on your website so people know. So the number of transplants are certainly increasing in the country. This year we just had our transplant group meeting last weekend, and for the first time we crossed 2,000 transplants in the country in a year. Thalassemia would have got a major boost from around 250. It's very likely that we crossed 300 last year. We don't have the up-to-date data. So it's very, in terms of percentage change, when you go from 250 to 300, it's not a big number, but the percentage change is very significant for a complex treatment like this. But in the larger sense, it's still nothing. And that's the message I told you last year, that allogenic transplant will never solve the public health part of this problem because the numbers are just too high. And therefore we need an alternative, and that was the gene therapy. This is just a concept. If you have a person with thalassemia, you take the blood stem cells, the way I showed you for hemophilia in the other lentiviral method. These blood stem cells, you can do 2 things. What I told you, what's being done right now in clinical trials is the lentiviral vector-based gene replacement. The lentivirus vector is capable of infecting the blood stem cell. That is why for thalassemia, people have chosen that vector. They can package the gene into the vector, infect these cells in the laboratory, make sure that the infection is adequate and stable, and then use the same stem cells to transplant the patient, autologous transplant. As opposed to what I just told you a minute ago, it is allogenic transplant, where you find somebody who is matching with you, your own brother or sister, some other member in the family, or somebody in the community who is fully matching with you, matched unrelated donor, and then do an allogenic transplant, which is a very, very different situation from doing an autologous transplant. Allogenic transplant can have major complications, and we know that anywhere in the world, even in thalassemia, somewhere between 1 and 10% of patients may not make it very well. And among those who make it, they may have some chronic complications in 5-10% of patients at least. As opposed to that, autologous transplant can have less than 1% mortality and almost no transplant-related major long-term effects, particularly if we learn to reduce the conditioning regimen. So there are some issues to be solved. So this is one part which is in the clinic right now. This part is the genome editing, and we had a small mention of genome editing here. This is being developed. It's not yet gone to the clinic, where you can repair the gene or turn on certain genes. So I won't talk about this anymore, but next year maybe if I'm here, I'll tell you the work we are doing and the world is doing with genome editing in this area. Because all thalassemia children are born with normal hemoglobin, with hemoglobin F. Right? You all know that. And for reasons science has never understood, nature decided to switch off hemoglobin F by around 6 months of age and then allow adult hemoglobin to come. And in thalassemia patients, that adult hemoglobin doesn't come. So by 6 months of age, you start dropping your hemoglobin. So if we can somehow just switch on the hemoglobin F, life should be good. And this genome editing is largely focused in the thalassemia sickle cell situation in switching on the hemoglobin F as one option, as opposed to the lentivirus where we are not messing with the hemoglobin F, We are just putting a normal gene and trying to make enough hemoglobin. Okay. So here, we will skip this. So the first report here was in 2010, where they took a patient with E-beta thalassemia. So they were playing safe. You know, E-beta thalassemia runs between 5, 6 to 7, 8 hemoglobin. You need to make a huge amount of protein to actually have successful thalassemia transplant. So here they were not sure how much protein will be made, and look what happened. So just again, this is a repeat of last year, but let me explain because they may be different people. This is where the transplant was done, but these red dots are the blood transfusions. So for almost 1 year, 8 or 9 transfusions were done, and then after 1 year, there was hardly any transgene. The red bar is the hemoglobin that will come from the transgene, the gene therapy gene. In the first year, there was hardly any. It's only after 11-12 months that you see the red bar. And the red bar finally goes on to make about 2-3 grams of protein, of hemoglobin. So from a baseline of 5, 6, 7, you add 2 or 3, okay, it is still significant. And that's when they reported it. Things have moved from there. And about 2 years ago, they showed that based on changes they made in the vector and the transgene, they can now get about 5 grams of transgene hemoglobin. So even if you take a thalassemia major, and that's what they have shown here, the beta 0, okay? These are the severe thalassemia, right? They're getting about 5 grams. So severe thalassemia may have baseline hemoglobin of only 2, 3, 4, 5, something in that range. If you add 5 to that, you can reach a level where your transfusion requirements can almost stop or become very less. And that is why this is not yet reached in the world a situation where they can ensure that you get at least 6 to 10 grams of hemoglobin expression. So even with this gene therapy, will everybody become alright? Well, the data is good so far, because even the thalassemia major makes 3, 4, 5 grams. Less than 3 grams are very rare. So you add another 5 to 3, 4, 5, you are in a very safe range where you are without transfusion. So that's what's happening. This has gone to phase 3. Phase 3 is going on, and you see the people who are asking me, can they go to Thailand? And you know why it's happening in Thailand and not somewhere in India maybe? Why is it happening in Thailand and not India? Anybody knows? No laws. It's very simple. So the man who started all this is a French person called Philippe Lebouche. So you look at the publications, the basic studies. He's a scientist, he's not a physician. Philippe Lebouche did all the preclinical work, and then he set up, he was part of the company, the Bluebird Bio. And then when Bluebird Bio got the first results, its stocks went up into 1 or 2 billion. Maybe Dr. Chandru will know more about the value, I don't. But it went up very much. He became so rich. And you know what happens in France if you become very rich? 75%. Tax is 75%, above a certain level. So naturally the very rich run away from France. So, but that's a different category. You don't have to be rich, you just loot and run. But this is the honest rich who want to run away, right? We were not attractive enough as a tourist destination. So Thailand was much more attractive, okay? So that he lives in Thailand, Philippe Lebouche. So naturally, he collaborates with the center there, because actually the clinical end of this is not a big deal. And you know how these studies are running. So if a patient is here, you take the stem cells like we do for many diseases, and you transport the stem cells to their laboratory. It will travel there. They will do this transduction. They will infect it with the lentivirus, with the transgene. They will check it and make sure it is good for transplant. They will send the cells back, and then the transplant is fairly routine, absolutely, straightforward to do. So that's, in Thailand they are not doing the transduction. They are a center where the patient can go. They will take your stem cells, send it somewhere. It will get treated with the vector, and then you'll get a transplant. Okay, so that's how it's going on. There are many other trials also going on from other groups, but these are the most advanced. So again here, it's expected that in about 2 years, the phase 3 trial will be completed, and if it's successful, which is very, very likely, there will be a product. Question is, what will be the cost? The thinking in the community, as you can see the cost, nobody expects it to be less than $500,000. $500,000 will be considered a deal, okay, for them, not for us. So again, I told you last year that we've been working with Emory. We have several transgenes that we are evaluating. We had tested it in the cellular model last year, showed that it is expressing hemoglobin. This year what we did is to take it to the mouse model, right? So the mouse model, what we did, again, very similar to what I told you earlier. You take the mouse, you take the stem cells, you transduce it with the transgene, then you irradiate another mouse which is similar to this one, and you put the cells back and see whether it is making hemoglobin. Okay, so that's been done, and what this shows you that by about 3 to 4 months after the transplant, these mice are making enough transgene hemoglobin. So that's the first step. Now beyond this, there are no, this is not taken to the non-human primate. So we need to work on this over the next 1 to 2 years, and then it is actually theoretically ready for a clinical trial. What will be a challenge? So anyway, with regard to thalassemia and sickle cell disease, gene therapy is here. Phase 3 starting means the product will be here soon. How do we get this to us? We have some challenges. So, the vector production, there are issues with technology, transfer. There are many people who have some experience in this area, but there is no group that has actually done it before in this country. There may be intellectual property issues, but I think that's less of a problem here because our Supreme Court is good and our laws are good. There is forced licensing, so if somebody makes it and is life-saving, we don't have to worry about those things too much. But because of that, technology transfer is not easy because people are worried in the West, if they give it to us, we will run with it maybe in a way that may not be good for them. Then the issue of, when we propose a trial, and this is something where I want community involvement a little bit more, and the societies, you have to make enough noise that just like we heard from DART. So DART has been or is going to get approval for a trial. Like that, this is a big challenge. So I told you last year, I didn't want to repeat the story, we made a proposal for a sponsored gene therapy trial for hemophilia in 2004-05 in India. That was the Avigen trial that was running from the US. 6 months after we made the proposal, the trial stopped in the US because there was some very minor toxicity, and at that time there was a lot of sensitivity about that toxicity in the liver. Nothing happened to the patient was okay. But we didn't hear from our regulator, the DCGI, for 2 years. So this, knowing that, we now, got this committee established and we have laid some grounds. But there's a lot of inertia within that system that doesn't move in the right pace. And I think a little bit of pressure from the patient groups, both hemophilia and thalassemia, saying if there's anybody in the country who's trying to do gene therapy, then please make sure that these reviews happen rapidly. We are not saying don't review, don't critique. You must review, you must critique, you must ask for the right things to be done. But the bureaucratic delay of holding the meetings and giving the responses should not be a block in the process. Okay? Then of course we have a challenge also at the DCGI level because there isn't that expertise in the country on how to evaluate applications like this. So this is again something that we need to work on. They have a cell biology based therapeutic drug evaluation committee. That is CBBT Tech. That's the forum where something like this will get discussed. It exists, it needs to do. Then if it goes to phase 2-3, at least for the phase 1-2, the government has given funding. Phase 2-3, where will the funding come from? And the discussion that was being done, you take it straight to the patient from the lab, there are challenges. Who will then take it beyond that? You see, there is no drug without profit. So somebody has to be convinced enough, some industry, has to be convinced enough that you will invest in making this drug, whether you sell it, no, that's greed. Okay, selling it for 1 million, half a million is greed. But they are not in our healthcare system. They are in the healthcare system where either the government or the insurance will pay. In Europe it's the government, in North America it's mostly the insurance, somebody will pay. But in our system, if you take something like this straight from the lab to the patient, if all the ifs and buts have not been sorted out, or most of the ifs and buts have not been sorted out, which industry is willing to invest on this unknown entity? Because they have to cross DCGI, they have to meet certain barriers. So it's a complex situation, and we can discuss this after maybe the talk, but it's not easy to say you are in a rush and I can understand the desperation. I can understand the human aspect of this, but it still has to go through certain phases. It can go very fast, and there are examples of what Dr. Kiran Majumdar said of 10 to 12 years is the usual number. There are examples of innovative drugs going from bench to the clinic in 1 or 2 years also, if we do everything right and fast. And the kind of collaboration that was being mentioned by the Karnataka initiative is a fantastic thing, so that's the kind of thing that moves things fast, where everybody is around the table and you get things moving. So I think, some of those things we need to address while the scientific work is going on. So I may have crossed my time, I don't know, but I am done. So that's all that I want. I want to thank people who are all involved, various people in this, but particularly colleagues from Emory in Florida. Actually they do all the preclinical work for nothing. I'm only trying to make sure we contribute towards the GMP, production of the product that we are doing the phase 1. We prefer to do it there for the phase 1, and then bring the technology here with an industry in India. And I just want to remind you that our next Cell and Gene Therapy Symposium is on 6th and 7th. This is for the moment only medical and scientific, but hopefully this will move to a Cell and Gene Therapy meeting, scientific meeting, where we will have both scientific, medical and lay people through a society or something like that. I'll stop there, and if you have any questions, I will try to answer.
The patients have a lot of questions.
No.
Hello there, doctor. What kind of support can the patient? What kind of support can the patient communities can follow in order to speed up the process? As you said, the patient communities need to put in process, some kind of a pressure in order for it to be going, right? What kind of a pressure we could put in as a society, as patients, or as united front? What would be the better approach to go ahead, and how do we go ahead?
So it's at each level of the process development, right? So one is fund more people to do this research in India. Okay. 2 is those who are doing research, ensure that it is moving in the direction towards the clinic, and make sure the reviews for those are done in time. You can get involved in them. That concept has to come into this country, where patients are involved in the decision making towards research and treatment. It's not there right now, but I think the country's landscape is changing. The understanding and involvement of patients are changing, and they should be there in these bodies to push for what is important for them. So specifically, support for research, faster reviews at the scientific and regulatory level, and then at the political level, awareness that this is happening in the world. This is a huge public health problem in this country. And right now, it so happens that, so it's no secret that Mr. Modi is seriously interested in sickle cell / thalassemia, given his commitments he has done in Gujarat. And there's a lot of focus, the whole CSIR, the Council for Scientific and Industrial Research, all the CSIR 6 or 7 institutions have a major program started 2 years ago on sickle cell disease. It's that focus. So that political pressure, that not just at the central level but the state level, people should be talking and looking for ways. And then you can now say, well, gene therapy has come somewhere in the world, get it here. We don't care it costs $1 million. We want it.
There's a wonderful story about patients with breast cancer in the US. So, way back in the 70s when Nixon had declared the so-called war on cancer, and the National Cancer Institute had been set up and so on. Patients were kind of given the impression that there was a solution around the corner, right? And nothing much was happening. Main things that were being used were chemotherapy, radiation, so on. So there was one survivor who was a lawyer, and she was actually a really powerful sort of speaker and so on, and she started organizing the breast cancer patient group, and she made a big sort of public outcry about why is it that research is not producing anything new. And finally it went to the US Congress, and then they actually came up with a plan. They actually came up with a plan and said, we need targeted therapies. We don't want this just chemotherapy that's a sledgehammer, that is all this side effect, and that makes us really suffer and so on. So why isn't there a more targeted approach? And scientists at NCI rubbished. They said it can't be done. But they prevailed. The patients prevailed. And the Congress said, yes, we should fund this plan. And the proposal was actually quite ambitious. In those days, it said it needed $300 billion to bring out a targeted therapy. And then the Congress didn't have a budget for it. So the Department of Defense, in fact it was the Army, which had unspent money. Maybe they didn't buy some tanks or whatever. So they said, we will put 210 million. That's all we have now into this. And they put that money in. And, the National Cancer Institute sort of reluctantly followed, right? But all the way, it was the patient community and the fact that defense had got involved, that they forced a certain discipline on the research and said, you will translate, you will look for solutions. You can't just say it's a heart problem and shy away from it. Right? And what came out of that was Herceptin, which was the first, it was the start of the whole idea of personalized medicine and targeted therapies, and of course it had all been developed with public funding, but then it was licensed to a company which became Genentech, because people at UCSF had been involved in this research. They started Genentech, and so that was the birth of this whole. So again, it's a very important story because it was really driven by the patient, and there are many such stories. And I think actually, right? We have DART, we have several agencies, but I think the thing is this thing about being able to roar. I think and that's what Kiran was also pushing this morning.
So it's more challenging for a situation like that where there is no success yet anywhere in the world. So you have to discover for the first time, and that's more challenging. And yet there are things happening. But what I want to suggest to the patient community, that's the majority of people sitting here today, keep track in the world what's happening. And if it's happening, then it must come here. It can come here in two ways. Either you buy the final product or you develop it here. Don't tell me you will not do either of these two. You have to do one of these two. Okay? And China is the best example. You know, look at these CAR T cells. These CAR T cells are the most wonderful thing that could have happened in terms of cancer treatment. And there are more CAR T cell trials with in-house, well, in-house is the wrong word, with China-made CAR T cells. There are more companies in China making CAR T cells than in US. Okay? This is not only going to help the patients there, it's going to be a huge industry for them. And we missed the boat completely. 4 or 5 years ago, you know, I suggested to various people, look, this is happening, we should get into this. There's not one lab in this country that has made CAR T cells so far. People are just starting thinking. So, like that, you know, you must keep a watch on the world, what's happening, and demand one of these two. Either bring the product or develop it here. So,
Dr. Srivastava, a very nice talk and nice comments. There's only a sort of general question with regard to your comment on technology transfer from the West to a country like India. And this is something that, again, we should keep talking about and see how to get around the problem of those players outside of India developing and not transferring because of the other issues that we have. A similar thing is happening in the ag biotech area with regard to the BT cotton. So seed industry have got together and are literally killing each other and shooting themselves in their feet, right? So the companies who developed, put in years and years of work and money, they're not getting their technology fees is almost gone, price control, and now the prices of a packet of seeds coming down almost every day. So now it's so much to the extent that the company that has developed this technology is not transferring any more technologies. So there is resistance of pink bollworm that we only keep making noise about. But no one in India is even attempting to do any work on how to combat the pink bollworm resistance. So we're not doing this, and we're not in a position to invite the technology. So if something like that would happen here, I think we're going to be in a huge danger. So we really need to talk about it.
We've got so many problems. One is to bring BT cotton. We've already got so many farmers suffering. Why do we need to bring that kind of technology?
But
No, the problem with BT cotton is that.
Let's not get into that.
No, because you made a comment. I need to respond to that.
And I will try to explain. I will try to explain. So, you know, we, I have tried to deal, you know, I know that the biotechnology department funding will get us through the phase 1, hopefully. But beyond that, we need industry. That's the model in the world. So we've been talking to some of the major industries in the country. Most are not interested because they would rather play the safe, proven cancer, you know, biosimilars or generics, where money is assured. This is the less traded area. Nobody knows what the market will be. Those who want to be in it, even if we go to university. So the collaborators, you take Emory or you take Florida. For that matter, you take even the published one, non-India, outside. So the first hemophilia trial was by Amit Nathwani, he is an Indian origin person, based at University College London. He does the research in collaboration with St. Jude. They have the publication. His research is now owned by his institution, and the Office of Technology in his institution sells it to Unicure, licenses it to Unicure. Unicure is a company based out of Netherlands that now is developing it as a gene therapy product. Amit is out of the equation. He can't use his own research anymore to do anything with that at a cost that he can control. So here, Florida, you know, Alok Srivastava is the scientist there, no relative of mine. He has done the vector work. Trent Spencer in Emory has done the transgene work. The university owns the licenses now, owns those IPs. And they will license it to the companies that give money for that license. That's the challenge. When we want to officially bring it to an industry here, they are sub-licensing for big money. Industry here is not ready to pay that kind of money. How do you solve that? So I tell you what the back route to this is. The back route to this is this. You look at the IP filing. Actually, they have not included India or most of these countries in their IP filing. So we are not in the zone where they can claim IP. But the scientific expertise has to somehow come. Through this collaboration, these scientists are knowingly giving it away. They don't care. Let the companies make money in the West. If this expertise comes here, then of course the industry has to feel safe taking on something like this, which is not protected in our territory and therefore can be taken forward. And for that you need the industry, science, academia collaboration with trust in each other that it can actually go forward.
All I'm saying is that I think we should be able to, if we're going to, either we put in a lot of effort to develop our own technologies to make sure that we are not dependent on any foreign powers, or if we're going to bring those technologies because we are not putting enough time and money into this, make sure the technology developers are appropriately listed.
Yeah,
That's the problem.
yeah, absolutely, yeah. Sir, patient, please.
You have answered partly about the sub-licensing. Let's say it's been given, how much time it will take for coming to market? Let's say industry has been started, how much time it will take to reach the public? Point A. Will the cost be same or how low is going to be? Third, will our genotype will have any impact because that's a Western. Another trial study is to be done in India again. So,
not at all. That last question is much easier to answer. So your genotype is anyway a wrong gene here, right? And we share the mutations. There may be some different mutations. Doesn't matter. We're not repairing anything here. We are putting a new functional gene that will function in a Caucasian as well as it will function in an Indian or an African or anybody. Okay. And already, in fact, some of the patients in the hemophilia trial done in the UK were actually from South Africa. Okay, the physician there was very clever to send them to, South Africans can get into UK much easier than Indians. Okay, so they went there, they got the gene therapy. So that's not an issue at all. In gene therapy, that's not an issue at all, so we don't go there. The other two questions are more difficult. It's more about business and economy, and I'm not the most qualified person to answer that. But let me tell you this. The moment the trial goes to phase 3, it always has an industry sponsor. Very rarely will academia take it to phase 3. Phase 3 means that they have built capacity for the expected market because there is money at the end of that. Industry needs money. So they will not do a phase 3 unless they have built capacity to actually market as soon as approval comes. Because the rush in phase 3 is to recruit the patients, get the results, submit it to the regulator, get approval, market. That's the race. So now, is industry in India ready to do that? That's a big challenge for us.
There's another question.
Doctor, this is a layman's perspective or like whatever we read from the internet. So hemophilia is something which is the gene therapy, whatever is done, it has to be, couple of genes has to be rectified. In thalassemia, it seems only one gene that needs to be rectified. So in that case,
so hemophilia, these are all called monogenic disorders. In hemophilia A, it's factor 8 gene. In hemophilia B, it's factor 9 gene. In thalassemia, it is beta globin. In sickle cell also, it's a beta globin, but a specific, one mutation. In thalassemia, it could be different mutations. But the bottom line is, we don't care what mutation. We are putting a normal gene. It doesn't matter what you have.
okay. But the thing is that the hemophilia thing, we have the success route for hemophilia is much more progressive than thalassemia. That's what we want to understand. Why is it?
Because AAV, so in hemophilia, nature gave us AAV and we somehow discovered AAV. And AAV is finding its way through a vein injection. So you know, the 2004 trial that we had proposed, when Avigen was doing it, we had to take the hemophilia patient, do an arterial catheterization, and inject the AAV into the hepatic, the liver artery, you know, the blood vessel that supplies blood to the liver. So it was a very invasive procedure, not a big deal. You know, the interventional radiologist will do it in 10 or 15 minutes. But it was invasive because science did not know that if you simply injected the AAV into the vein, it will find its way there. That was discovered later. So now AAV is harmless and it is a peripheral vein injection. If you can make the vector, choose the right patient, you can treat him in this room and gene therapy is over. Right? As opposed to that, thalassemia, you have to harvest the stem cell, take it to a clean lab, do the transduction, make sure that the transduction process has infected enough of the stem cells so that those stem cells will now behave with the normal gene and make enough hemoglobin. Plus the amount, see, the first patient that was reported in 2010, he made only 2 to 3 grams of hemoglobin. It was good, it was success, something was coming, but 2 to 3 is not enough. Most of the thalassemia major will only become an intermediate if they make 2 or 3 grams. They will not become more than 8 or 9 grams and be near normal. So thalassemia has been a challenge from the point of view of the type of treatment needed, from the cell in which to express, take out the stem cells, manipulate it outside. Then even now, see the model, the day the hemophilia product comes, the product can come, you can keep it in the refrigerator, freezer of your pharmacy, patient comes, you thaw it, you infuse it, it's over. Here right now, the cells have to be taken, sent to a lab somewhere that is equipped to do the transduction, then they have to send the cells back. Then you have to do an autologous stem cell transplant. So it's, you know, much more involved. But, you know, if it works, it will be good.
Thank you.
I have a question.
There's still one more.
So, like bone marrow transplant, is there an age restriction in gene therapy that by certain age or earlier the better kind of a thing?
Not really. You know, so we can derive, you know, this answer from what is being done for autologous. See, autologous transplant is currently done mostly for cancers. In a disease like multiple myeloma, it's a protein-making cell cancer, plasma cell cancer. You are doing autologous transplant for 70-year-olds. So autologous transplant can be done for 30, 40, 50, 60-year-olds, there is no problem. Technically it's the same, whether we do here, only difference is, you know, there we take the stem cell, keep it out, nobody is manipulating it, we give intensive chemo and give the cells back. Here we take the cells, send it somewhere, hopefully do it in your own place, and then put it back. But the rest is the same.
My question is, I think you said that once the institute or the university does the research, then it becomes the property of the institute in the West, and then they sell it to a private company, and you know everybody profits, but the person who actually made it can't send it out. Now the question is that in the West, a lot of the university funding, especially if it's state or even private university, comes from either NIH or other sources, and then DARPA, you know, also funds some of this research. In India, you have DBT or I don't know if ICMR supports industry. Now, suppose you are trying to bring in at a lower cost. What is the scenario right now? You're saying industry has to come in at a particular stage to do it. Now, how many industries here? Because now, if you take a Gilead medicine for Somalia or something like that, it's sold at like $94,000 or $89,000. When it came to Asia, and because a friend of mine actually works in Gilead, he demanded that it should be sold at a lower price, otherwise he's not going to work on it. And you know, they started offering it in India at a lower price. It's still unaffordable for a large majority of our people, you know, even if it's subsidized. So how is it the system here, and how do we as people, because is it state public money that's being used for this research, and then what happens, because I know you're trying to bring this at an affordable rate, so you've been discussing it with my husband at different stages. So how does that work, because the large companies are going to come in regardless and say we want profits, and when the small company comes in and has to struggle again to find the funding, KBIT, what is it called? Kibitz has this 29, little startups in the incubator. Somebody is funding them, they're giving them space. A lot of money is being spent on infrastructure from what I saw in that presentation. How much of it is actually being spent on giving these companies actually research funding, It's more in infrastructure space.
So I'm not sure I'm the right person to answer that question. Probably Chandra will give you a better answer at home today. But
if you follow the news media, there's always been a lot of grants that have been announced. Not huge grants, but definitely matching some of the DBT grants when it comes to startups. There's idea to be oversee 50 lakh, right? And there is elevate additional support. Something like our minister has claimed that something like 400 crore money has gone to multiple startups, right? And there is TBIs that have been established in multiple places, engineering colleges and so on. Apart from what we are giving to the incubators in DBC. So there is a lot of money going into, but we've also put in the new policy document that Vijay probably knows, where we need to be supporting R&D in the area of all the emerging areas like rare diseases, stem cell research, medical devices, marine biotechnology, and so on and so on. But again, this year budget has been cut because there are so many other things for the government to be looking at. So this way we have to be roaring more and more and more in how strong our case compared to someone else's case. I'm not saying every money that is going to every other area is justifiable, but this is where we need to fight together, with all the communities coming together and really roar very, very loudly.
No, I think the expenditure on the infrastructure and so on, which has been developed, is actually very well justified. I think, to tell you the truth, I think with BBC infrastructure, without that infrastructure, none of those biotech companies could actually move forward because of the central instrumentation facilities and so on that have been provided there. So I don't think we should be talking about misspent monies. I think the bigger issue is how this whole path of commercialization takes place. That comes much later after the startup sort of mode. And there, for example, in the US, you had this whole thing called the Bayh-Dole Act. Right? The Bayh-Dole Act essentially said even if research has been funded by the state, when an invention actually happens, that intellectual property, the state relinquishes its claim to the university, to the institution where it was developed, and the investigator. Okay? And then they sort of decide what is the commercial path forward. So this in the US was in 1985, and it was sort of done for the land-grant universities. And after that, that has become sort of the norm. In India as well, I think, although Kapil Sibal was not able to push it through, in principle, all the funding agencies, DBT, etc., are open to the idea of the university and the investigator taking the IP forward together. Right? At least that is my...
So, if I understood your question right, it is not the challenge of the university or the investigator not receiving compensation for whatever work they have done. That's not the issue. They will get whatever the law or whatever the agreement is, 1, 2, 3, 5%, something in that range. The issue is more the industry that licenses it. Will they sell it at $1,000 or at $10,000 or $1 million? That the university or the investigator has no control on. If they sell at 1,000, these fellows will get 50 cents. If they sell at $1 million, these fellows may get $500, right? It's the industry that then decides what the cost is. And the philosophy of industry deciding the cost is very simple. What's the maximum that the market can sustain, pay? Right. It's got nothing to do
But that's
with...
my question, so
that's the
how,
issue.
when the government says that 50 lakhs, that doesn't get anybody anywhere to reach the stage to bring it at an affordable cost. So what does the government have to do to bring that gene therapy at an affordable cost where the major industry or already the multi-billionaires just want to keep, they have plenty to put in and will make more profit.
So we didn't
where do we get that small company that can make it and has the heart to bring it at an affordable cost along with the inventor?
So what we see is that, of course, we can be, taking a small company all the way to the end. But the hope is that small companies will come through some breakthrough innovations, and the industry will come to handhold them to take it forward, which is what happens in the US. But
okay, any other
I want to ask a question on behalf of the patient community. All the TAL patients are here sitting, but they talk in WhatsApp. But when it comes to asking questions, it's left to few of us to ask. On behalf of everybody, whatever was discussed, I'm going to ask. So BMT has a whole lot of exclusion criteria. And now the demographic divide of the thalassemia community across India is majority adults. Now, we want to know if gene therapy has any exclusion criteria, like some patients have Kella antibodies, autoantibodies, HIV, HCV, so endocrine complications and a lot of comorbidities. So that is the one thing that is worrying a lot of adult patients. And I want to ask this question: will there be any exclusion criteria?
So I tried to answer that partially earlier. Age-wise, it will not be, okay? Because people even 60, 70 years old with malignancies are going for autologous transplants. The challenge will be if there is a major organ dysfunction, like the heart, liver also, because you're taking stem cells out of the patient, you are transducing it with the gene, vector, and now you want to put it back. So you want to make sure the existing stem cells are fully destroyed. Because if these fellows stay back, they will dilute what you are putting in. There's only so much space in the bone marrow. If half of those cells are the untransduced, which didn't go out, so you have to destroy the existing stem cells fully.
That's with chemotherapy.
With chemotherapy, right now, okay? But if you see, people are coming up with antibody, with toxins, where you can just give a CD34 tag with a toxin and kill most of the CD34 cells that are left behind. So if we get to a conditioning where we are not using very strong doses of chemotherapy, which may affect the organs that are dysfunctional and make the patient very sick, then the exclusions will reduce. But the exclusion is only related to existing major organ dysfunction. So right now the patients who are going for gene therapy, they should not have heart for sure, because that's a very big challenge. And of course they should not have any major liver or renal dysfunction. But renal is anyway very uncommon in thalassemia. Liver can be an issue. So it all depends on how we can then reduce the conditioning. That is happening with antibody-based conditioning as opposed to chemotherapy-based conditioning. But other than that, the HIV, HCV, hepatitis B, those will not be a significant issue.
So the recovery time will be faster than the recovery time?
So, in autologous transplant, it takes about 10 to 15 days for the blood counts to rise. It will be similar. Because we have manipulated these cells a bit, it could be 2-3 more days, but that's not expected to be a major challenge. As long as it happens before 20 days. If you remain with low blood counts for more than 15 to 20 days, your chance of getting serious infection is very high. But if it's less than 15 days, it's no issue at all. If it's less than 20 days, it's reasonable.
This is that community. If you guys have a question, please ask it. Because you have been asking questions in the group. Do not expect that only me, Namita, or certain people are going to ask questions. So if you have questions, you have the chance now to ask.
I think many people have questions not about gene therapy, but about regular management of thalassemia.
So maybe what we can do is close the.
She has a question.
Oh, please.
Ask it. She wants to know when will gene therapy come to India.
I wish I could give you an answer. I am a little bit more sure about hemophilia at the moment, but thalassemia, where our challenge is to See, when you say come to India, the phase 1 will help only 5 or 10 patients. So come to India is only when it goes beyond phase 3 actually. So
How much does it cost?
no, right now there is no cost because it's still in trial, so nobody knows. But anywhere in the world there is no cost. It's a trial everywhere. So there is no product for gene therapy anywhere. You can't buy gene therapy anywhere in the world today for thalassemia. If you are included in the trial, then you will be treated. If you are not in the trial, you have to wait. So there is no cost.
Apparently, Bluebird announced in India that it's going to cost 2 crores.
Cost 2 crores. Huh?
That's what she said.
Maybe, yeah.
In Delhi, I think they have a meeting in Delhi. I think it's last year.
No, last year I was there in the meeting. There was no such announcement. Philip LeBouche was there. I spoke after Philip LeBouche and told him that we seriously want to have an indigenous option because we know that Bluebird cost maybe too much.
Where did you get that figure? Island Bluebird.
Yeah, I mean it could be something in that range. It's possible. But go back to Gilead. It's nobody's gift.
Right.
It is nobody's gift. It is the fact that the pharmaceuticals in India would have made a generic for sure, and so the company had a choice of either doing what they call compulsory licensing, and that's the most wonderful thing in the law in this country. You either do compulsory licensing or you give up your right to, you can't deny human beings life-saving drugs.
I mean, as an industry body, we actually presented to the DCGI saying that for orphan drugs, from now onwards, the country should seriously look at invoking compulsory licensing if we really want to address the rate.
But Chandru, compulsory licensing in the biotech arena requires scientists who are seriously engaged with it. And that's the tragedy of this country. And I'm saying this, that is what has showed up in CAR T-cells. China has got CAR T-cells coming out of every door.
There's a recent article of, I think about 80 patients have already benefited.
Indian patients going, yes, right. I was at this meeting where Indian patients are going to China for CAR T-cell therapy, for cancer.
So I think maybe we wind up the session and
Mariam.
because I think 6 o'clock was more or less.